This video for CFA Higher Chemistry will cover proteins and how they hydrolyze into amino acids and how intermolecular forces can affect their shape. So these are the learning outcomes we're going to look at. We're going to look at how proteins can hydrolyze into amino acids and also how intermolecular bonding can affect the shape of proteins. When we digest the proteins that we eat, specific enzymes in the body will break down the proteins into amino acids in a hydrolysis reaction. During a hydrolysis reaction, the peptide links between the amino acids in the protein chain are broken down to amino acids. One of the things you will need to be able to do is, if you're shown the structure of a section of protein, you'll need to work out how many amino acids were joined to form that structure, and also if you broke up that structure in a hydrolysis reaction, you would need to be able to draw the structural formula of the amino acids that would result from that hydrolysis. Remember, hydrolysis means to break up using a water molecule. So if we were to hydrolyze this peptide link here, we would need to put an OH back on here to generate the carboxyl group and an H on here to make the amino group. So in turn, we would end up with our amino acids formed from that structure. This structure shows a section of a protein containing 3 amino acids. What I'd like you to do is get a pencil and paper and draw the full structural formula of the amino acids produced from the hydrolysis of this section of protein. So pause the video, have a go, and then I'll show you how to do it. The first step in determining the amino acids that form this protein section of protein is to identify the peptide links. So the peptide links are shown here. Once you've identified the peptide links, we need to break the bond between the C double bond O and the NH. This is the bond that breaks when we do the hydrolysis. Because we're putting the water molecule back in, we need to put a hydrogen atom on the nitrogen atoms, and we need to put an OH onto the carbon atoms to obtain the structure of the amino acids. So we can do it with this molecule on the left, and the middle, and the right hand side. And now we have the structures of 3 amino acids. Here's an exam question for you to try, so pause the video and have a go. So when you break down proteins completely into small water-soluble molecules, you can see we have amino acids being formed. Pause the video and have a go at this question. The question is asking which of the following could be produced by partial hydrolysis of X. Note the words partial hydrolysis, not complete hydrolysis. There are 2 peptide links present in the structure. If we were to do complete hydrolysis, we would break this link and this link. However, it's only partial hydrolysis, so we're only doing hydrolysis at this peptide link or this peptide link. So let's start off with this one. If we break this peptide link and we move this part over here and put the OH back on to form the carboxyl group, and we attach the hydrogen to form the amino group here, if you look at this structure will match structure A. Pause the video and have a go at this question. So we're being asked for the structural formula for one of the molecules that would be made when this section of egg white protein chain is completely broken down. So we're breaking down a protein chain, so this is going to be a hydrolysis reaction. So the first thing we need to do is identify the peptide links. Once we've identified the peptide links, we need to break the bond between the NH and the C double bond O and put the H's and the OH's back on. So there's our peptide links that we're going to break the bonds of, and then if we're putting the water molecules back, we need to put an OH back on to form the carboxyl groups and the H's back on to form the amino group. So we've got our first molecule on the left, We can do this for the next one by putting the OH and the H back. And these are the molecules that would form from this hydrolysis reaction. So any one of these amino acids shown here would be an acceptable answer. These red pieces of plastic are going to represent amino acids that are going to join together in a condensation reaction to form the start of a polymer. So if I join these two amino acids together, we'll get a covalent bond being formed. The thing about protein chains is they tend not to be linear. The proteins can fold up into various shapes that are unique to that particular protein. So you can see I've got my amide link present in one amino acid. If I was to move things around a little bit, like this, and let's say that this chain had a amine group coming out here. Now this oxygen will be slightly electronegative because it's more electronegative than this carbon here. And this hydrogen will have a positive charge because of this electronegative nitrogen. So between these two atoms we can have an intermolecular force. In this case it would be a hydrogen bond. And this hydrogen bond can cause the protein chains to fold together in a unique shape. So I'm going to use this piece of plastic to represent a hydrogen bond. And what you can do is allow the protein to form a kind of spiral shape like this using hydrogen bonds. So I can coil this round again. Stick in another hydrogen bond, so this could be between an amino group and a C double bond O. And you can see they've got the start of a kind of spiral shape. And if I was to have a longer chain, you could end up like this, in which the protein chain forms a helix shape. And we call this an alpha helix. And this shape is found as a 3D structure in various proteins. The thing to note is that we have hydrogen bonds between the different amino acids in the chain, giving you this shape called an alpha helix. Here's another example of a shape that can be formed by a protein chain. So this is our protein with all the amino acids joined up by peptide links. And the chains are held together by the intermolecular forces between the chains. So these are not representing covalent bonds, these are intermolecular forces such as hydrogen bonds. So it's the hydrogen bonds or intermolecular forces such as hydrogen bonds that hold the protein together in these specific shapes. So hydrogen bonds would be an example. And the name we give to the structure is a beta sheet. It's a kind of fairly flat, rigid structure that's often found in proteins. This is a model of a protein produced by bacteria that binds to antibodies as a defense mechanism. You can see we've got our alpha helix here with the hydrogen bonds holding that structure in place. We've also got a couple of beta sheets with our hydrogen bonds there, and this is our protein chain going along here from one end to the other. So scientists can look at the 3D structure of these proteins in order to study its function and how they work. This is a 3D animation taken from the Protein Database Bank website, in which you can see the 3D structure of the protein. So as you've seen in the models we looked at, within proteins, the long chain molecules form shapes such as spirals, sheets, and other complex shapes. The chains are held in these forms by intermolecular bonding, i.e. hydrogen bonding, between the side chains of the constituent amino acids. So if we have a protein chain of amino acids, hydrogen bonding will occur between parts of the chain to give this helix shape, formed by intermolecular forces formed between the NH and C double bond O groups. As well as helix shapes, you can have sheet structures, which are formed by intermolecular bonding between the groups of the amino acids present in the protein chain. So if I have a protein chain of amino acids all joined together by peptide links, you can have this sheet structure containing hydrogen bonds which are formed between the C double bond O and NH groups present in the protein chain. As you can see here, proteins can be a mixture of helices and sheets and various other structures to give proteins their unique shape that allow them to perform their specific function. So if you take a protein in our body, it will have a specific shape at 37 degrees. This would be maybe the optimum temperature if it was an enzyme. However, if you heat the protein up to high temperatures, what will happen is the intermolecular forces between the protein chains will break because these bonds or forces are a lot weaker than the strong covalent bonds or peptide links between the amino acids. So what happens if you break these intermolecular forces? So if I break the forces between this alpha helix, you can see that the protein starts to lose its shape, and therefore it won't be allowed or able to do its function. And if I break up the beta sheet as well, then it's going to lose that structure as well. So in the end, we've got a pretty loose, shapeless polypeptide chain. So we say the protein is denatured and has lost its shape because we have broken the intermolecular forces between the side groups of the amino acids in the protein. So we know that each protein is a unique shape held together by intermolecular forces such as hydrogen bonding. However, if you change the pH and the temperature, this can change the shape of the enzyme and what's called denature it, and this stops the enzyme from working properly. So here's an example of an enzyme activity against temperature pH graph. So typically enzymes will work well, say it's our body, it would be 37 degrees. That would be the best temperature at which it works at. However, if you heat these enzymes, say above 60 degrees, then we get a decrease in activity or function because the enzyme has been denatured and lost its shape. This is because the intermolecular forces that hold the shape of most biological enzymes are broken around 60 degrees or so. When proteins are heated, intermolecular forces such as hydrogen bonds are broken, allowing the proteins to change shape and become denatured. If we look at this protein structure on the left, we can see a sheet and a helix formed by hydrogen bonding and intermolecular forces between parts of the protein chain. Upon heating, these intermolecular forces are broken, causing the protein to lose its shape and become denatured, and therefore lose its function. One thing to note is that the proteins are not being broken down to amino acids. We're not breaking peptide links or covalent bonds. You can see that the protein chain is still intact. All that is being broken are the weak intermolecular forces between the protein chains. Another thing to note is that the denaturing of proteins in foods causes the texture to change when it is cooked. So taking an example of egg white, it goes from a clear liquid to a white solid when heated. Pause the video and have a go at this question. The first question is asking, state the term used to describe the change in shape of enzyme molecules. And the correct answer for this would be denaturing. Second question, suggest a change in conditions that would cause the enzyme molecules to change shape. There'd be two acceptable answers to this. It would be a temperature increase or change in pH. Note that you have to say temperature increase. Temperature on its own is not acceptable. So anything suggests that you know that a higher temperature than the optimum temperature is required to denature a protein is also acceptable. Pause the video and have a go at this question. So this question is saying that during cooking processes, the proteins become denatured as the protein chains unwind and the egg white solidifies. Explain why the protein chains unwind. Well, the reason they're unwinding is because you're breaking the hydrogen bonds or intermolecular forces. So that would get you the answer. You wouldn't get the mark if you talked about peptide links being broken and covalent bonds being broken or any sort of hydrolysis reaction. This is a summary of the learning outcomes covered in this video. So if you've got this far, thank you very much for listening.